Great, thanks very much. So my name is Yates. I'm a lecturer in the Department for Mathematical Sciences, and today I'm going to try and answer the question of what is mathematical biology, because I'm a mathematical biologist. I often think it's useful when trying to answer these sorts of questions to try and think about what mathematical biology isn't. So in particular, it isn't this terrible joke about square roots, or it's a maths joke, but really, more seriously, it isn't about numerology, it isn't about looking at patterns and saying, oh look, there's a curve, it must be some sort of Fibonacci sequence. Fibonacci seems to be the only thing that people have ever heard of with mathematical biology. There's no reason why an elephant's trunk should be a Fibonacci sequence. So what mathematical biology is about is looking at patterns in nature, yes, and it is about trying to describe them mathematically, but it isn't as simple as just looking at it and describing it. We have to build a model, we have to try and make predictions about what actually happens and see if we change something in our model, how would that change the biology, and we go and test that with the biology. So these are some of the things that I work on. So these are looking at the way that eggshells get their patterns, looking at the way that locusts swarm and with devastating consequences, looking at the way that parasites can evade the immune system, and today in particular I'm going to tell you about the way that cells can migrate in the early embryo. So this is what mathematical modelling and mathematical biology for me are all about. It's this paradigm. We make a model of the biology that we've observed, we make a prediction, and we go back and test our prediction with the observation and see if it's right or wrong, and go back and change the model. So to give you an illustrative example, I look at modelling neural crest cells, which are a subtype of cell in early embryos. They're important for a variety of different tissues in the adult, and these neural crest cells, they try to migrate around from the back of the early embryo round to the front. So it's sort of as simple as that. At the same time as they're trying to migrate round, the domain is changing underneath them, the carpet's moving. So these two different stages of embryo look very different morphologically, different shape, but actually what's more stark is perhaps the fact that they have a very different size over the time period that we're interested in. So I look at a subtype of neural crest cells which are called melanoblasts, and they're responsible for the pigmentation in your hair and in your skin. So you find if you have a mutation in the gene called KIT, ironically for me, that the melanoblasts don't make it round to the front of the embryo from the back, and you get these white belly spots in humans and in mice. So we try to build different models of this mathematically. I look at two different models. I look at an individual-based model where you take each individual cell and put it into a computer and give it certain properties, and you look at how that density of cells evolves. I also look at a continuum model where I just model the density of cells, sort of take a step back and look at a macro-scale type model. So they have their advantages and disadvantages. The individual-based model accurately represents the biology, but it's slow to simulate. The population-based model is much faster, but you don't get as much detail. So this is the individual-based model. You take your domain, split it up into a number of boxes, and you position cells on those boxes or intervals as you choose, and you allow them to jump left or right out of the boxes into neighbouring boxes with certain rates. You can soup this up and make it more interesting, so you can add a signal gradient, this yellow curve here, which the cells can sense and respond to. So you might try and climb up this signal gradient, which is what they're doing here. You might add cell proliferation, you might add death, you might add various density-dependent movement phenomena. In each case, we try to build an equivalent density model from the individual one. You can also do growth, so you choose an interval to grow, it pushes the intervals along to the right, along just enough to put a daughter interval in, and then you divide the cells which are in that parent interval evenly between the two daughter intervals. Okay, so that's the model. Then we can go away and make a prediction from that model. So the red curve here is my predicted density of cells over time, and the blue histograms here represent the density that's actually been found in an experimental setting. And you can see that actually it doesn't match very well, which is sort of what we're looking for in mathematical biology in the first stage of the cycle. So we start with 100 cells at the bottom there, and actually, you know, if we changed that and made that far fewer cells initially, say 10, then you see that you get a much better fit to the experimental data. Okay, it's still not perfect, we still need to tweak some things, but it's much better. So we go back to our experimental colleagues and we say, can you just test again, just to be sure, how many cells should there be at the start? And they go, yeah, we should have done the test properly at the start, you're right, absolutely. And it turns out there should actually only be around 15 rather than 100, which was the original number. So we actually get a much better prediction, and our model has said something about the biology. So that's just one cycle through this model, predict, observe cycle that we do in mathematical biology. And sometimes the observations are wrong, as in this case, but sometimes the model is wrong. We have to go back and tweak it and make predictions and then check them against the observations. So this framework is a way of actually saying something quantitative about biology, and for me that is what mathematical biology is all about, using this framework to try and describe the amazing patterns that we see in nature. Thanks very much for your time.